For this video, I'm going to cover A2 topic 14, homeostasis in mammals, and I'm going to focus on the gross structure of a kidney, and I'm going to describe the structure with reference to the structure of nephron using photomicrograph and electron micrographs. So before we go into the detailed structure of a nephron, I'm going to introduce you with the structure of a kidney to see how does the kidney looks like. Therefore, only we understand where is the position of the nephron. So kidney in general, we have two main layers to focus on. This layer is known as the cortex. And the layer inside will be the medulla. And part A is known as the pelvis. And structure B will be your ureter, where this is whatever that is going to be excreted will be delivered via the ureter to the bladder, and before they are being secreted out through the urethra. How about the arrows? What are the arrows showing? So the arrow going in obviously is taking the blood in to be filtered. So this will be the renal artery. And the blood that is coming out from the kidney will be carrying the blood that is being filtered, will be known as renal vein. So, what is the outermost layer then? This one is known as the capsule, but usually it's not being mentioned because it's not involved in the process of filtration. So where does the filtration occurs, or where is the filtering unit? So the filtering unit in a kidney is known as a nephron, and nephron, it's usually shown as, you will see like a U-shaped structure here, a small one, known as the Bowman's capsule. And you have all this convoluted tubule, known as proximal convoluted tubule, and a bigger U-shape known as the loop of Henle, and another convoluted tubule known as distal convoluted, and the collecting duct. So pelvis is where all the collecting ducts are collected. Therefore, all the waste products will be collected and excreted via the ureter to the bladder. So if the question is asking, where does the ultrafiltration occurs? We know that near the Bowman's capsule is where we have the branches of the arteriole forming the glomerulus, and we have all these smaller blood capillary. So the filtration will be occurring at cortex. So U will be the cortex region, and the loop of Henle will be found mainly in the medulla, which is the L. And the last one will be which area the blood urea concentration is low. So pay attention, the question is asking about the blood urea. Therefore, it's either renal artery or renal vein. So since they're focusing on low concentration, therefore it must be renal vein. How many nephrons do we have in a kidney? For each kidney, we actually have about millions of nephrons to filter our whole body's blood. So now we're going to look at the flow of the blood within the nephron and the associated name of the blood vessels. Number one, we're going to start with renal artery, as we know that renal artery is going to deliver blood into the kidney. Then the artery is going to branch out into smaller artery known as afferent arteriole. And as you can see, the afferent arteriole actually has a larger diameter. Therefore, it allows pressure difference to be created as the efferent arteriole will actually have a smaller diameter. This contributes to the pressure needed for ultrafiltration. Then as the efferent arteriole branch out to form capillary, it will form a knot of capillary known as glomerulus. And this is where ultrafiltration is going to occur, where the filtrate is going to be collected in the Bowman's capsule, or here shown as renal capsule. And the filtrate is known as glomerular filtrate. So after the blood is being filtered, they have low water potential as they are mainly red blood cells and large plasma protein remains. And this blood vessel will be known as efferent arteriole. So what happens here is the efferent arteriole will need to collect all the useful substances. Therefore, they will branch out to form capillaries surrounding the PCT and the DCT. So I'm going to show the extension of the blood vessels. So from the arteriole, you will usually see blood capillaries surrounding the PCT and DCT. And moving downwards to the medulla region, surrounding a loop of Henle and the collecting duct, before they return to the blood circulation via renal vein. So let's look at the role of each blood vessels. So after ultrafiltration, that's where we're going to move on to PCT. So what is the role of PCT? PCT is in charge of selective reabsorption, where all the useful substance will be reabsorbed, such as glucose, amino acid, ions, and majority of the water will be reabsorbed here. So this blood vessel that's surrounding PCT and the DCT is known as peritubular capillary. And moving into the medulla region, we have blood vessel known as vasa recta. This blood vessel is involved in reabsorption of water from the descending limb and from the collecting duct. After ADH has decided how much of water to be reabsorbed and the water potential of the blood is returned to set point, the filtered blood will be returned to the blood circulation via renal vein. So now we're going to analyze the cross-section of the cortex. So as you know, in the cortex region, we can see the glomerulus, the Bowman's capsule, the PCT, and the DCT. So from this diagram, how do you distinguish all this structure? First of all, try to find an island-like structure where it looks something like this, where it's actually separated from the surrounding cells by a space. The space is very significant as it will most likely represent the Bowman's capsule space, where the glomerular filtrate will be collected. So as you know that if there's a space, the island-like structure with lots of blood capillary, where it's significant that you can see some red blood cells, that will be known as the glomerulus, which is this middle part of the structure. So I'm going to shade. So this is the glomerulus. And And what forms the glomerulus? You have the wall of the blood capillary. So immediately near the glomerulus, the outer layer, this would be your endothelium. And outside this blood capillary, actually it could be the podocyte. So endothelium or the podocytes, which forms the ultrafiltration barriers. And how about the outer layer then? On the outer layer of the Bowman's capsule space, that is also the epithelium, but it's the outer wall or the outer layer of the Bowman's capsule. And moving on, what are the tubules that are surrounding your glomerulus? So in the cortex, we will be able to see PCT and DCT. So what are the adaptations of PCT and DCT? For a PCT, they actually have smaller lumen as it has brush borders. If you zoom in, you might be able to see microvilli. So what is the purpose of this? To increase surface area for reabsorption. Therefore, this structure we can identify it as a proximal convoluted tubule. In short, it will be known as PCT. It's not very significant, the microvilli here, so I'm going to show the extension. And how about DCT then? DCT has a thinner wall as it doesn't have brush border. So this is distal convoluted tubule, and the drawing, sometimes it's shown like this. You will see that it has a thinner wall and therefore a larger lumen. This will be the photomicrograph for the cortex region. So what can you observe if you're given a cross-section of a medulla region? So we know that in the medulla, you can observe loop of Henle and collecting duct. And how do you distinguish the cells then? So collecting duct cells are actually cuboidal. That's how we describe the structure. It's cuboidal epithelial cells. So the feature would be a bit cube-like structure. This will be the collecting duct. And for loop of Henle, the cells are flat and thin, only showing the nucleus that is prominent. This is known as squamous epithelial. That's a feature of the cells, but it's known as loop of Henle. The other one is also known as loop of Henle. So what's the difference between these two? One actually has thicker wall and the other one has thinner wall. As we know that the descending limb actually has thinner wall and the ascending limb will have a thicker wall and is impermeable to water. But sometimes we might not be able to distinguish, but at least you can tell that if it's squamous in shape, it's actually loop of Henle cell. And finally, the last one is actually, you can see some red blood cells inside. That's the blood capillary. The understanding of the structure of the kidney is very important before we move on to the function and the role of each part of the kidney. So if you're interested to know more, do not hesitate to contact me for group or private tuition. Thanks for watching.
That's all for today's video. If you're interested in more genuine sharing by other geniuses,
please subscribe to our channel and don't forget to turn on the notification bell, ding dong. Also, if you're struggling with one or two past year questions and the mark scheme just doesn't seem to help, Genie has got you covered. Feel free to let us know what question it is by filling in the Google form linked in the description below. GenieUs Hub will get genius teachers to fulfill your request for the solution. Genie will see you next time. Bye-bye.